Hello, I'm Jennoa Fleming.
And I'm Beverley Buchanan.
And we go to Open High School in Richmond, Virginia. The title of our project is The Effect of Hydrilla Verticillata on Species Diversity in the James River Rock Pools.
Invasive species have huge economic and environmental impacts on the areas that they inhabit. In the upper left is an image of a lionfish. It is native to the Pacific Ocean and threatens reef ecosystems in the Atlantic. In the upper right is a picture of the emerald ash borer beetle. It is native to Asia and threatens ash tree populations in the United States. In the lower left are zebra mussels, which are native to Russia and Ukraine and clog water treatment facility pipes in the United States. And in the lower right is an image of hydrilla, which is the aquatic plant that we studied in our experiment. Here in this image, it has formed a large mat and is caught in a boat motor. Hydrilla is native to Asia and it was introduced to Florida in the 1950s through the aquarium trade. It is found in 30 states throughout the United States and is both asexual and sexual methods of reproduction, making it hard to contain. The United States spends millions of dollars annually to control its spread.
The questions we wanted to answer with our experiment are what factors contribute to the distribution of hydrilla and whether or not hydrilla has a negative impact on biodiversity. For diversity, we looked at species richness, which is the count of different species in a certain region, and species abundance, which is the number of individuals per species.
Here's our study system of the James River rock pools. The James River rock pools are located on Belle Isle just west of downtown Richmond. Here's a closer view of the rock slab where the rock pools are located, and they're indicated by the numbered markers. And here's an even closer view of an individual rock pool. On the right is an image of dragonfly larvae and physis snails, which was collected during a sample.
So our first hypothesis was if hydrilla is present in the James River rock pools, then it will be more common in larger pools close to the river. We came to this hypothesis because the river is a way for hydrilla to get into the rock pools, and we thought that the closer and the larger the rock pools were, then the more hydrilla it'll have. Our second hypothesis is if there is hydrilla in the James River rock pools, then it will reduce the richness and abundance of the species in the rock pools. We came to this hypothesis because hydrilla takes up a lot of space and could overcrowd the rock pools. Over the course of our study, we compared pools with varying amounts of hydrilla to pools without any. We used research gathered from 152 rock pools from the summer to the fall of 2017. We looked at overall species richness, how far the rock pools were from the river, and what effect the presence or absence hydrilla has on snail populations. To collect data, we chose a rock pool and recorded physical factors that could influence our data, such as size and distance to the river. We also recorded biotic factors such as canopy coverage and hydrilla coverage. For hydrilla coverage, it was recorded based on 3 categories. None was 0%, some was less than 50%, and a lot was more than 50%. After recording these, we swept the pool, recorded the species type and the number of species that was in the sweep, and then we emptied the dish back into the pool and repeated this 9 times. This is an image of students from the MSI summer program sampling a rock pool. And this is an image of what was collected from a sweep by a VCU undergrad.
So the first thing we looked at was relative distance to the river. So we found that in pools with and without the hydrilla, the average distance from the river was about 4 meters, so about the same. And we looked at relative size of the rock pool and found that there was no difference in rock pool size with and without hydrilla. We next looked at overall species richness, and in 3 different varying categories of hydrilla, we found that there was no statistical difference indicating that varying amounts of hydrilla did not affect species richness in the rock pools. And we looked at physis snail abundance, in varying levels of hydrilla and found a statistical difference between the none and some categories and the some and a lot categories, whereas the none and a lot categories were not statistically different, indicating that some hydrilla increased Physis snail abundance. And we looked at planorbid snails, where we found a similar trend where some hydrilla increased planorbid snail abundance in the rock pools. So to conclude our experiment, we found that pool size and relative distance to the river was not affected by the amount of hydrilla present, and we found that species richness was not affected by hydrilla, but our research did show that the average number of Physa and planorbid snails was significantly greater in areas that had some hydrilla.
Our results showed that rock pools with some hydrilla was beneficial. It may be beneficial because it's a primary producer, it makes oxygen through photosynthesis, and provides a food source and a habitat. Too much hydrilla could be harmful because it overcrowds the rock pools, blocks out sunlight, and threatens organisms that need space to survive. We also observed while sampling rock pools clear capsules. Through research, we found that snails use hydrilla to attach their eggs on, so we speculated that these clear capsules could be snail eggs. This may be another reason why hydrilla may increase snail abundance.
So the finding that hydrilla can be beneficial is in contrast with much of the research done on invasive species. We think that more research of how snails use hydrilla can help us understand the relationship between these two species.
So we would like to thank Dr. James Vonish and the undergrad students from VCU and students from the University of Richmond. We would also like to thank the students who participated in the MSI summer program and our biology teacher, Emily Betts.